Thank you very much, Linda, for that very kind introduction. Really appreciate it. And it's a pleasure for me to be here this evening. It's morning for you, so it's an honor as well. And hopefully I can come there in August to be there to co-present with my colleague here, Richard, for maybe 1 or 2 workshops for the ISPE group there. So with no further ado, I will go ahead and start in on the workshop here. And at any time at the end, we'll have around, hopefully around 20 to 30 minutes for a Q&A session. So I'm going to go ahead and start in here. And today, the workshop we are going to be focusing in on disinfectant validation and efficacy testing. And this relates to the products that you use in your clean room and the validation of them to be used on the different hard surfaces, hard non-porous environmental surfaces in the clean room. So the agenda is going to be, I'm going to be speaking and taking a deeper dive into disinfectant regulations. So both how the products are regulated by the manufacturers, and then how the industry, so biopharma, medical device, cell and gene therapy are all regulated by the regulators relating to disinfectants. Then I'm going to be covering some of the ins and outs of manufacturers and what's required to actually register a product globally in Europe and in the United States. I'll be covering some of those testing methodologies and what is necessary to get a registration. Then I'll be talking a little bit about how the testing lays out for end users. So these are our pharmaceutical, biotech, medical device, compounding pharmacies, and cell and gene therapy, which in Europe they call them ATMP facilities, need to do in terms of validation of these products. Then I'll be covering a little bit on some recent regulator observations. So what is the FDA saying? What is MHRA saying? ANVISA, some of our global regulators. Then Richard's going to be taking the reins, and he's going to be covering disinfectant coupon testing, all the ins and outs of that, because that's a critical step. And then I'm going to come in at the end to take it to the touchdown yard line, where I'm going to be covering field trials and the in situ validation of these products, which is a really critical step these days, especially for our industry regulators. So that being said, let's get into it here. So we have our first poll question here. And Linda, are you guys asking the poll, or am I? There it is. Okay, good. Do you want me to read it, or is someone reading it on your end?
Go ahead, Jim.
Okay, I wasn't sure. So with this poll question, the question is, what has your facility completed to validate the sanitizers, disinfectants, and sporicides in your clean room operations? So have you completed coupon testing, the in situ field trial validation, both coupon testing and field trials, just the routine environmental monitoring, or are you simply relying on supplier data? And now you have the chance to go ahead and take your answer, and then you can submit it. And then I think we get the results real time here. Am I right about that, Linda?
Yes.
Okay.
I think you might be able to see the results coming up.
Okay. Let's see. Oh, wow, voila. So it looks like most of you, 36% of you, are relying on environmental monitoring data. That's a good thing. You should have that. And then 28% are doing coupon studies. That's good. Only 11% are doing field trials, and only 13% are doing both coupon and field trials. And then around 11% are relying on supplier manufacturers' data. So environmental monitoring, that's a good thing. Coupon testing, and it looks like I'm going to have my work cut out for me taking the deeper dive with all of us into field trials, because that's important too. So we can go on from this. I guess it just disappears. There we go. All right, so in the US, all of these products we're talking about, so sanitizers, disinfectants, and sporicides, are all registered in the United States actually as pesticides with US EPA. And the EPA actually has a website, and it spells out for you how to get a product registered. So when we come up with a new formulation, there's a number of things, and it costs quite a bit of money to get that formulation registered. And so what does EPA do? And EPA, by the way, stands for Environmental Protection Agency. So they would approve the active composition. You have to have a registered active with them, such as quaternary ammonium or a phenolic, in order to register the formula as a disinfectant, for example. You have to complete what is called the 6-pack of safety testing. And so that means it'll be toxicity data such as inhalation tox, dermal toxicity. They'll be looking for LD50 data, and there will be these 6 different toxicity tests that you're required to complete with the formula and share that data with them. Then you're going to have to put on there what the directions are for use. So is it something that I can wipe on a surface, spray on a surface? Is it something that I can fog? How is it going to be applied to the surfaces in the room? Is it mopped? So you will be coming up with the language that is going to be on the label there and submitting that for approval. Then one of the most critical things is the efficacy data. So you've got to generate data at an outside contract lab against the different bacteria, viruses, and molds and yeast that you want to have on the EPA approved label. And that data has to be reviewed by EPA and approved to get it on the label, which means then you have claims against the specific virus or bacteria under the use conditions. And so where that testing comes from is there's a big Bible-like book, and it's a gray book, and it's called the AOAC. AOAC stands for Association of Official Analytical Communities, so it's a giant book. And I believe it's chapter 6 of that pertains to disinfectant testing. And it spells out the methodologies for sanitizer tests, disinfectant tests, and sporicidal testing. So that is these AOAC tests that I mentioned right here. And you can see here one of our operators in our micro lab actually running a test. So this is basically a little porcelain penny cylinder. It's coated with spores that are dried on there in a desiccator. So it could be a 10 to the 6 concentration of Bacillus subtilis. And then it'll be placed into a tube with broth in it and neutralizer like you see here. After that hard surface has been exposed to, let's say, a sporicide or a disinfectant for a certain period of time, maybe 10 minutes. So the test is what we call a pass-fail test. So the sporicidal challenge test, you'll have 720 carriers. They will be porcelain penny cylinders and silk suture loops, which will simulate porous and non-porous surfaces. And you have to show complete kill on all of those surfaces. So you can't have any of these tubes show up positive. So that means you can't have any turbidity in the tubes after they've been exposed to the disinfectant or the sporicide. And that is why I say it's a pass-fail test. If you fail it, then you're not going to get the claim. So these regulations that we talk about, the key thing with US EPA and the European, which is European normative testing, is that you need to show that that disinfectant is effective at destroying and killing the microorganism, whether it's a bacteria, a virus, a mold, or a yeast, right? And so in terms of the vendors, so our company is a manufacturer and a vendor. We have to generate this test data against the specific microorganisms and show that we're able to kill that organism. Now, when you generate efficacy data, meaning pharma, biotech, med device, you are going to use representative coupon surfaces from your clean room and they will be inoculated with the organism, and you're going to have to show kill on those surfaces. So that's how your testing will differ. So where's some guidance in the industry on the testing? Here are some of the guidance documents out there. One is the new EU Annex 1. We are actually all the way up to draft 13. And sometime, hopefully, during our lifetime, hopefully by the end of April or May, the final version of the draft of Annex 1 will come out. And what it actually states relative to disinfectant efficacy testing is it says, validation studies should demonstrate the suitability and effectiveness of the disinfectants. So it really doesn't give you a whole lot of guidance. The current PICS document, in terms of validation, what it states is when you test disinfectants, you should be going for the minimum wet contact time on the different surfaces. So those are a couple of the key guidance tools out there in the industry for you. Another good one is the FDA's Aseptic Processing Guide. That's from 2004. And that talks about the suitability, efficacy, and limitations of disinfecting agents and the procedures for testing them. So it does give some general guidance here. And this document, we think once Annex 1 is finally approved and ratified, what we believe in the US is you'll see this aseptic guide come up, and the regulators and the industry will be taking a look at it and updating it and revising it as well. And then there's actually a new revision of the USP 1072 that's floating around out there in the Pharmacopoeia Forum. Tony Kendall was one of the key authors of it. So he and I have been talking over the past few years, and it has updates to the log reduction numbers for molds and yeast in it. So it'll be nice when it actually comes out to confirm some new things. But the current version of it does talk again about selecting suitable disinfectants and verifying their effectiveness in terms of surface challenge against the microorganisms on selected surfaces. And it talks about log reduction numbers. So we'll get into that. Then there's PDA's Technical Report number 70. And that also provides, there's about 2 or 3 pages in it dedicated to the validation of disinfectants. It does get into qualification of antimicrobial agents, so sanitizers, disinfectants, and sporicides. It has a nice table in it, incidentally, which I updated today, and will be submitting the new table into PDA to approve it and basically modernize it so that it mimics what we see in the new version of USP 1072, because the table currently in it on log reductions is, in my opinion, a bit outdated. So we want to make sure that they are in sync, right? Here's a recent 483. It's a quite good one that came out from the FDA. And I thought it would be helpful here to kind of highlight it and talk about it, because these are the things that our Food and Drug Administration and other industry regulators, such as ANVISA, Chinese FDA, MHRA, HPRA, the regulators in Australia, all of these regulators are looking at things like this when they get into your efficacy testing. So here they talk about, first of all, the whole study, if you look at the bottom here, it's over 10 years old. So when efficacy studies get really old, 10, 15 years old, they may be older and antiquated. And so the test methods may have advanced and changed since they were run. The log reduction numbers that are required today may be different than they were back then. And you may have new surfaces, new microorganisms that certainly should be assessed from a risk assessment, risk analysis standpoint, even what I call a gap analysis, to see, do I need to do some additional testing and include those new organisms in this? So looking here, as I said, it's 10 years old, so it's really kind of outdated and antiquated. There were no in-house environmental isolates used. That's a big problem, because one of the main things you're going to hear from FDA regulators like Thomas Arista, John Metcalf, Sharon Toma, good friends of mine in the industry, is how effective are these products against your environmental isolates? Well, we don't know at this particular site because they were never tested. So that's a big, big issue. Then they come in here and they say under B, the study included no other surfaces present in the clean room. So they didn't have some of the key surfaces in the room that will be highlighted. That will be a concern, right? And then they talk here about when they looked at the microbial populations tested, it said it consisted of spores or dividing vegetative organisms. So that it says here at the start of the sentence that there was no analysis to determine what organisms were actually tested. So is it a bacterial spore? Is it a vegetative organism, right? That's important to know that. And on multiple occasions, they say here, we used a disinfectant in the gowning room. However, there was essentially no study to evaluate the bioburden in that gowning room. Was it even included? And it says under E, the method of cleaning the clean room is different than the method used in the validation study. So there may be even some call into question of, well, am I using mopping and wiping in the room? How do I assess that? I mean, am I just going to be looking at how effective the chemistry is wet on the surface? That's an important concept. So you need to, when you run these studies, you need to have scientific rationale, as Thomas Arista says, for what you're doing. Here's another good one, and this is a few years old, but I think it's a really good one. It came out of the Southeast US from an FDA regulator. And one of the key things they're all looking at are the coupon surfaces. You can see here, they highlight the coupon surfaces, and it says many of these were not representative of the surfaces found in a tissue processing lab. So my guess is the regulator walked in there and he noticed that we've got, you know, new wall surfaces, maybe a new epoxy flooring. There could be some different types of stainless steel, maybe 304, that were not part of the study. When things like this happen, you're almost always going to see either a warning letter or a 483 come up. And I can guarantee you that. I see that quite a bit. And the regulators are trained because they come through the Parenteral Drug Association training courses where they will see me or someone like me typically that teaches a course on this where I will be highlighting things like this because my goal is to educate them. And they should be looking at this if they get into, have a reason to get into and look at your validation studies. So here are some of the key things the regulators are looking at, and this is not just the FDA. This is MHRA out of the UK, HPRA out of Ireland, Chinese FDA, the Australian inspectors, ANMAT, ANVISA. All these regulators are looking at things from your sanitizer disinfectant studies and sporicidal studies. One of the key things is they want to see how effective these are against your environmental isolates. That's a key thing. It's fine to have what we call reference strain testing against environmental type culture organisms. Many times your manufacturers may have some of that data, but it's really very important that you have generated some data against your EM isolates. They highlight here that many times the data does not support the appropriateness of the disinfectant. I see this a lot of times at compounding pharmacies where all they end up testing is IPA. And what is the problem with 70% IPA? Does it kill everything? I don't think so, right? Doesn't kill spores, we store them in it. And this is another big one, not using representative surfaces. So you should be doing a full-blown risk assessment with the surfaces from your clean room facility, and then assessing, you know, what surfaces need to be included in this study, what can I exclude? What is, and you should be looking at this from the standpoint of what's worst case, what's most commonly occurring, is the surface in a high traffic area or a high risk area? And that may give you an idea of, you know, do I want to include that in my study? You definitely need to include environmental isolates. If all you've tested is reference strains such as ATCC, which is American Type Culture Collection, you're really missing the boat because regulators want to really see this kind of data, right? And you should have data that supports the wet contact time. So how long do I need the product wet on the surface to kill the organisms? And that's generally 10 minutes for a disinfectant and a sporicide. But with a sanitizer like 70% IPA or 70% ethanol, Maybe it's anywhere from 1 to 3 minutes or less than a minute, because keep in mind, when I spray a surface with a alcohol, it's not going to remain wet very long, right? So I always think this is a very helpful slide. Now that Annex 1 is talking about contamination control strategies, it's something I'm going to be publishing on. I'm working on an article and a PDA poster with Novartis to put out soon. And I think this is a very helpful thing to kind of keep in your state of mind, because it's all about taking a big picture look and a holistic approach of contamination control in your operation. So facility design, and one of the big things we talk about at the Parenteral Drug Association are aging facilities. As facilities age, things like the ductwork and the flooring and the sealant layer on the floor break down over time. Those can become a bigger and bigger potential risk for contamination and particle generation. Materials and how you pass them through into the clean room. That's always a big concern, a big risk. You don't want to pass something through that's got spores on it, right? Personnel, and that's your number one source of bioburden in the room. 80 to 85% of all the bioburden in the clean room comes from people, right? It's your movement in the room, operators, visitors, anyone walking around the clean room, and behavior. I've been to many sites where, for example, they put the SOP on the floor. I actually audited a site in the southern US where they kept telling me they keep getting hits from a canine bioburden and they couldn't figure out what's going on. And I take a look at the videotape, and about 2:30 a.m. in the morning, the security guard gowns up and he goes walking through the clean rooms with his German Shepherd. So I then decided maybe I should call Kimberly Clark about new gowning material for the German Shepherd. But thus, the German Shepherd should never be going through the clean room, right? I've been to clean room facilities where operators are eating in the clean room. That shouldn't happen either. Or smoking. Those things should never happen. That's all a big risk in terms of contamination. The New England Compounding Pharmacy that had the big issue in the US with molds and a lot of fatalities from fungal meningitis, they were turning off the air handling system at night. That's not a good idea at all, because mold just loves that hot, moist environment like you have there in Singapore and in Puerto Rico, right? So it just grows like wildfire then. Another big thing can be your process. So whether you're doing cell and gene therapy, where mold can be a big issue, or blood fractionation, where molds or Bacillus cereus can grow under cold room conditions. A lot of times, whatever process you're running can lead to either more or less contamination. Many times it's more. If you're running a tableting operation, so that powder can be a source of bioburden contamination. And then we talk about utilities. So water for injection systems, biofilms are a big concern there. Another big concern, there's an organism called Cutibacterium acnes. It's an organism that causes bad acne. So it's operators touching their face and touching a surface in the room. And this bug can grow under aerobic or anaerobic conditions, being a big issue and risk. So you have to look at that, and you have to look at compressed gases, helium, nitrogen, argon. Those are all potential sources of bioburden. Where you place equipment in the clean room. So if you ever block off the entry vents to the HEPA filtered air, that can cause clean room eddies in the room that spin around like a tornado and cause, bioburden concerns. So placement of equipment, placement of anything, that could block the filling zone or the HEPA filtered flow of air around the filler, that's a risk. So moving on to the next slide, this is my favorite slide of all, and I've actually had presenters in the industry ask me to use it, and I'm always fine with that. I think this is a great source of understanding, because when we look at this is a very delicate balance in the industry. So here at the bottom, we have disinfectants and sporicides, and why it's such a delicate balance is our pharma, biotech, med device, cell and gene therapy facilities, they have to balance in choosing a product. They have to look at what are the most important criteria in making that choice. So is it efficacy? Normally it's the key pillar. Compatibility, my friend Fred Ayers at Lilly would tell you he doesn't want to use any product that's going to destroy his insulin filling area. So compatibility can be a risk and an issue. What about stability? That's always a big concern too. How long is the product stable in a use dilution or in an open container? FDA actually looks for this kind of data now. Safety and disposal. So is it something I can put to the drain with running water? Do I need to neutralize it? Is it something that can go straight out into the wastewater stream? So those are all things. Is it something where I have to wear a respirator? So safety and disposal are a big concern too. And then another one can be rinsing. So periodic residue removal and rinsing so that you don't have a safety risk. You don't have a risk of the aesthetics of the room changing colors. So what are the key things that the end user has to be concerned about? So our pharma, biotech, med device companies, and cell and gene therapy, you need to be concerned about these 3 items over here, which is the in vitro testing. And my colleague here, Richard's going to get into this. He does a beautiful job. He's going to be talking about suspension testing and, more importantly, the coupon studies. That's something that you should do with the products you're using. And I'll be covering the in situ field trial data. That's another key element. And as 35% of you said, a lot of you have this environmental monitoring data covered. Why is that so critical? Well, if I were to ask our audience here, how do you know the product's really effective? Coupon data is great, field trial data is great, but your real litmus test is that ongoing environmental monitoring data. That shows, is it actually working in the room or not? So recommended procedures here. These are some of the best guidance tools currently out there in the industry. One is the USP 1072 chapter. As I mentioned, before the year's up, you're going to see a new version of it with some new log reduction numbers in it. There's an ASTM guides. Our company actually championed this document. It's very good too. There's the ISO 14698, and this is a document that we're actually going to be pulling out and updating in the next couple years because I'm on the ISO committee. I know Anne-Marie Dixon is one of our lead ISO representatives. We are going to be looking at updating that. PDA's Tech Report number 70, I actually made some edits to that today in the log reduction table that I'm going to be submitting for approval. But this is a great document. It's around 100 pages long. I would highly encourage you, if you have these kind of products you're using in your clean room operation or your controlled area, to purchase a copy of it. If you're a member of PDA, there's a different tiered membership they have now, and you can probably access this for free. There's also a IEST document. This is the group that's associated with the ISO documents, and I co-authored this technical report here. So I would encourage you to get a copy of that because it does drill down on how to actually do a 1, 2, and 3 bucket cleaning routine, how to do the mopping and wiping in a clean room, what direction, what coverage, and it's based on scientific data and research. So here are all the different types of testing in the industry. Your manufacturers are required in the US to do the AOAC testing. And again, that's pass-fail testing. In the US, Puerto Rico, and parts of South America, one of the most common coupon studies you'll see run is we call it the Quantitative Carrier Test 2. And it's an ASTM test method. It's E2197-17. And you would use representative coupons that are basically going to be little 1 centimeter coupons for the testing. There's also a glass method here, and that's 2111. The Europeans actually updated their method recently. They updated the section on fungal testing and the use of the glass beads for recovery step. And that method here, it's EN 13697. It was updated in 2019. So that's the most common method used in Europe. There are some French methods and German methods, but they are all supposed to be harmonizing to the EN methodologies. And the Australians have their own methods as well. I also want to mention in Europe, there is a wipe method. It's EN 16615. It has some room for improvement. They are looking at revising and updating it now with the EN committee. because currently you use this very large stone and you're going to be putting a wipe on the bottom of it and wiping across the surface. So it is even possible with the current version of it to get water to have a 2 or 3 log reduction. And they're looking at ways to improve that method. So here is one of the key methods. It's the EN 13697, updated in 2019. You'll see here in this container that the way the method is run, you put the 2 centimeter coupon in the container with broth, nutrient media with broth, like say TSB broth, and the glass beads are in it. So if this is my recovery step, I put it in there inoculated side face down so that when I shake it, I'm shaking the beads to hit it, and the organism should be coming off into the solution. So this shows here our inoculum. When I inoculate the surface with the micropipetter, I am going to be inoculating on there about 50 microliters of the microorganism, and then I'm going to desiccate and dry it. Then I come over here and I add the disinfectant or spore side. I add 100 microliters there and let it dry. And then I'm going to recover, and this is the recovery step over here. And I have a video of it if anyone's really interested. The US method is very commonly used, and not only in the US, you see other countries using it too. It's the ASTM E2197-17. Notice how much smaller the coupons are here versus the last slide. See, these are 2 centimeter, and the US method is 1 centimeter, very small. So here I micropipette on 10 microliters of microorganism, maybe Staph aureus, Bacillus subtilis. I then will desiccate and dry it. Then I add 50 microliters here of test product, so disinfectant or spore side. These are US pennies, so you can see how small these little coupons are, and you'll see a big size difference here. Again, see that. So I just wanted to highlight that. And a recovery step in this one is going to be sonication and vortexing, which are very good recovery steps. So one of the big concerns that we see in the industry here is coupon size. In the current version of USP 1072, which, as I mentioned, is going to be updated very soon by Tony, what we see is it recommending a 2-inch by 2-inch coupon, which is extremely large. So you're not going to be able to fit this in any container or beaker, which makes it extremely difficult to recover from it. So when I inoculate it with my micropipetter with organism and it dries, I'm going to have a very difficult time with a black light trying to locate the inoculation site. So adding disinfectant to it is complicated. One of the things that they've talked about in the industry to use as a recovery step with these very large coupons are stomacher bags, which is 2 paddles will hit this in a bag and you'll be recovering from it. The problem with the stomacher bags, that's something used in the food industry, a very crude method where you put meat in there to do a recovery step, just not very efficient and leads to some kind of erroneous results. But as I mentioned, one thing I do want to mention actually is the current version of the USP 1072. When I spoke to Tony about these huge coupons, he said they were never meant for coupon testing, that it's designed to be a wipe method only. So no one does that or understands that in the industry. So one of the big debates out there is the coupon size. That's a big debate in the industry right now. These very large coupons, as I just showed you, 2-inch by 2-inch, which is 5.08 centimeters by 5.08, are huge. Not easy to work with at all. So there's all kinds of different things mentioned out there. So TR70 talks about 3.8 centimeter or 3.8 centimeter by 3.8. Again, a very large coupon. If you look at the ASTM method, this is where we talk about the 1 centimeter disc. The European method, the 2 centimeter, and we have customers in Europe using the 5 by 5 centimeter up here, and we have some actually doing a 28 by 28 centimeter, which are so large, good luck recovering, right? So the larger coupons make it almost impossible to recover. And the key from a science standpoint is have something that's scientifically sound, and that's much more important than arbitrary size. Let me go back here. So make sure I didn't miss anything. So a key thing is when you as an end user, so pharma, biotech, med device, compounding, pharmacies, and cell and gene, you're going to have representative coupons as such that you see here that you're going to cut down to size to use in your studies. Some examples of the coupons you're going to be using will be 304 stainless, various plastics, Lexan, epoxy flooring, MMA flooring, vinyl flooring, terrazzo, and you will be doing a risk assessment on this. So you may start off with 30, but when you risk assess it and gap analysis it, you're going to hopefully get down to 5, 6 surfaces. So very low number. You might even test surfaces like nitrile or rubber gloves or gasket materials if they're common in the room. One thing I did want to mention is manufacturers like our company, we're required with the AOAC or ASTM tests to use very specific coupon surfaces. So this is very different. It's very regimented compared to what you will be doing here with your representative surfaces. So now we have the second poll question. I don't know if you want to put it on the screen and I can read it. Okay, this is a good one here. Has your clean room facility completed a detailed risk assessment, which I've been talking about, to determine which surfaces to include in the coupon study? Either yes or no. We don't have an I don't know here, so it's either yes or no. Now wait for you to put your answers in.
Jim, you have quite a divided room.
Oh,
50-50. It's like 42%
really?
yes and 48%
Wow.
no right now. We'll show the answers in a second.
Sure. That's good for us, Richard. We have lots of work to do. So, okay, the no's almost 60%. Wow, that's a big number. The yeses 41%. So yeah, that's a pretty distinct difference. So we'll work on that. We'll work on educating you and giving you some feedback in the industry and enlightening you to what's out there so that you can make an informed decision. Thank you. So I'm almost ready to turn it over to Richard here. Not quite yet, just about. I'm not trying to take up all your time here, Richard. Is this you, Richard, or you're not quite yet? I don't know. You tell me. I think. Let's see. I got these
25.
slides here. Okay,
yeah,
that's right.
Slide 25.
Yep, because I couldn't see the number. Okay, so let me talk about this. Neutralizer methods are very important because you have to stop the chemical reaction from killing the microorganism at a certain time point to mimic how you're actually using the product in the room. So the most common method for this is chemical neutralization of the active. So one of the easiest ways to explain this is if you have a product with hydrogen peroxide, you need to basically use 10 microliters of catalase to neutralize it. Otherwise, the hydrogen peroxide keeps killing. And at 10 minutes, I really need to stop that reaction. So adding the right neutralizer with that coupon study is critical. Or if it's a suspension study, you also have to neutralize it. Another method that can be used a lot in the industry for neutralization would be dilution. So you can make a 1:100 dilution with water for injection to neutralize the alcohols. Many companies will also do filtration where you separate the active from the microorganism. So some of your big biotechs will use this method and then rinse it. So with a rinsing agent. One of the key things is a watch out that you need to be careful of. Some of the neutralizers like thioglycolate, thiosulfate, which is used with bleach, can actually be toxic to the cells. You want to make sure that you're not artificially killing all your cells with the neutralizer. So that's why it's critical and something we talk about in PDA's technical report number 70. You need to validate the neutralization step. That's important to make sure the neutralizer is actually doing what it's supposed to do. Many of the products you're using, disinfectant products, there will be a specific formula to use to neutralize the active, right? So if it's a phenol or a quat, I'd recommend checking with the manufacturer. So this is a very critical slide that we talk a lot about in the industry. And on this slide, we show what are the most difficult to kill organisms versus the easiest to kill in the industry. So up here at the top, we have bacterial endospores. Some of them can be extremely hard to kill, and over at the right, I pull some of them out. So organisms like Bacillus cereus, Penne bacillus, Bacillus sphaericus, Bacillus thuringiensis. They have a thicker exosporium on the outside of the spore, which is a layer of sugars, which are very difficult to penetrate, making it harder to penetrate into the spore. Some of them also have certain proteins on the core of the spore, which act kind of like a repellent and can repel getting hit by biocides. So some spores can be very, very hard to kill. Not often, but occasionally, maybe every 10 years, we find one that is really in that trajectory of extremely hard to kill. So your sporicidal agents can typically kill, and that would be like your bleaches, your hydrogen peroxide peracetic acid, and VHP can kill from bacterial endospores all the way down this spectrum. Your phenolic disinfectants can kill from TB or Mycobacterium on down the spectrum. And your quaternary ammoniums can kill from fungal spores like Aspergillus penicillium on down the spectrum. And alcohols, like 70% IPA, 70% ethanol, they can kill from TB down the spectrum. The key is to get that wet contact time. So one other big debate in the industry is the pros and cons for doing coupon studies. One of the big issues and concerns in the industry is the industry wants to reduce the amount of testing that's needed and having to do additional testing every so many years, because it's a big resource and cost. It's a big cost center too. Some of these studies can cost $100,000 all the way on up to $1 million plus. So one of the ways that the industry has looked at being able to sidestep this is to create basically a big centralized coupon study that everyone can refer to. The BPOG group attempted this. So there is a PDA journal article that came out at the start of the pandemic that's out there. If you want a copy, I can get you one. But that document is out there. It talks about how to do risk assessments for a global disinfectant study. I think it's very useful, but it really doesn't accomplish what we set out to accomplish here. Another thing that they have looked at is to work with the PQRI, which is the Product Quality and Research Institute, which does do scientific testing for FDA. And one of the things they were looking at there, one of the big pharma companies was championing looking at manufacturers' AOAC data and kind of giving that to PQRI and saying, well, we really don't need to do any additional testing because this is worst case. And there's arguments that could be made like that. That's possible because those are very rigorous and hard to duplicate tests. So you could make that argument. But one of the cons for that is out there that really says you do need to do this testing, some organisms don't behave like the rest. So some of the spores, like Bacillus cereus, a bacterial endospore, and molds like Cotomium are much, much harder to kill. So you may require a longer contact time or additional methods, maybe a cleaner and disinfectant in order to kill it. And then we even have biofilms, which can be an issue too if you get them in drains or sinks. So in terms of acceptance criteria, this slide's important for suspension or time kill studies where the organism is surrounded 360 degrees by the biocidal agent. You should easily be able to get a 4 or 5 log reduction. It's easy to kill it. USP 1072 currently states a 2 log reduction for bacterial spores, 3 log reduction for vegetative non-spore forming bacteria like staph. The new version is going to have a 2 log reduction for fungal spores and yeast. And PDA's technical report number 70, I have updated today to match 1072, but the current version of it that's out there talks about a 1 to 5 minute contact time for disinfectants and sporicides, and that you need to achieve a greater than 1 log reduction. And then a 90-second contact time for alcohols as a sanitizer for greater than 1 log reduction. That I probably won't change much. One of the key things you need to do is evaluate the effectiveness of your disinfectants and sporicides. So here's some industry data here. We looked at the mold spore, Aspergillus brasiliensis, pretty hard to kill mold, and Bacillus subtilis. And what you'll see here is hydrogen peroxide peracetic acid, ready to use, and bleach at 5,250 ppm of available chlorine. Very, very effective sporicides. At 1 to 10 minutes, they kill my hard to kill mold. And at 5 to 10 minutes, or 5 to 20 minutes rather, you see pretty good efficacy coming out of them against the bacillus. However, 6% could take up to an hour to get the same kill. It takes a lot longer to get it going. If you started with a concentration of 10%, you'd see a lot better efficacy, but 6% is just not that effective as a sporicide. So I wanted to highlight that. And then we have done some testing against an extremely hard to kill here, bacterial spore, a strain of Bacillus cereus, which is serious if you get it. And pardon me, you'll see here that at 20 minutes up to 60 minutes, 60 is in the white, 20, and pardon me, is in the blue. So what you'll see, the hydrogen peroxide peracetic acid, fairly effective at the longer contact times, bleach at high concentrations. And what we ended up coming up with is a combination of bleach and hydrogen peroxide to be able to kill it. But the cleaners don't do much. The iodine containing disinfectant doesn't really touch it here. And then we had a concentrated sporicide that we used at 1 to 18, a little more effective. But again, the exosporium, we believe, on this particular spore made it extremely hard to penetrate and kill. So we need to have a two-pronged approach to kill it, with a bleach-containing cleaner and a hydrogen peroxide peracetic acid chemistry. So with that, Richard, it is your time to shine.
Okay, thanks, Jim. So I will continue from here. So this section is on the common causes of the failures during the coupon studies. So as you know, coupon studies can involve a lot of tests. And during the testing,
sorry, Richard, you're fading out there.
There may be some cases of failures. Causes of failures can be due to some general reasons. Sometimes it can be due to neutralization or preparation of inoculum, and also the condition of the surface, the coupons that you provided for the testing, and also the recovery of the microorganism at the end. So it's some of the general causes of failures. So one is the improper dilution of concentrated disinfectants. So you have a incorrect dilution ratio that may cause issues during the testing. And also if you use the AOAC test methods for the coupon studies, so this is generally not appropriate because the AOAC test method is basically a pass-fail test method. So it is not appropriate for the coupon studies. And also the ineffective chemistries. So if you have the, for example, testing alcohols or disinfectants without the sporicidal claims against the endospores, so you will get a failed results. So the bottom line is that you need to use a sporicidal agent for those spores testing. And also the contact time too short. I understand that there are some microorganisms require a shorter contact time than others. More challenging microorganisms such as the Aspergillus brasiliensis typically will require a longer contact time for the disinfectants. So you need to understand the contact time requirements for the disinfectants. Okay, next slide. Okay, so this is the neutralization issues. So when you perform the coupon studies, if you use a chemical neutralizer, so take note of the things that you need to know to prevent neutralization issues occurring. So one of the issues is the incomplete neutralization. So if you have an ineffective neutralization, you will have the non-dilutional series of the plates. So basically this will cause the inconsistent reduction factors during the dilution of the plates. And also the uneven distribution of microorganisms. So during the inoculum, you will need to spread it out evenly so that prevent the clumping of the microorganisms, which can affect the results as well. And if the neutralization is not effective, the results can be an overestimation of the activities of the disinfectants. And of course, during the validation of the neutralization, potential toxicity of the neutralizer will need to be assessed through verification tests. And one of the good guidelines on the neutralizer validation is the ASTM 1054. So this is basically the standard test for the evaluation of the inactivators of the antimicrobial agents. So this is a very good guidance documents on the neutralizer validation. Next slide. Okay, so these are some of the neutralizers mentioned in the PDA Technical Report 70. Of course, the alcohols, you can use dilution or polysorbate 80, and also quaternary ammonium compounds. For example, you can use the polysorbate 80 and lecithin, even the phenolates compound as well, polysorbate 80 and lecithin. As you mentioned, the hydrogen peroxide, if you have a hydrogen peroxide component in the sporicidal agent or the disinfectants, you will need to have a catalase because the hydrogen peroxide is also killing the microorganisms. Next slide. And then these are some of the more neutralizers which can be found in some of the guidance documents like catalase, lecithin, lithin, polysorbate. So you will need to basically choose an appropriate neutralizer for the disinfectants or the sporicidal agents that you will be testing, and perform the neutralizer validation to make sure that it is effective. Next. So the inoculant problem, so making sure that the bacteria is at the right phase of growth, and also the clumping of the inoculum. So clumping of the inoculum will pose the problems of not contacting with the disinfectants or the chemical agent. So in those cases, you will get failed results if you have a clumping of the inoculum. And also the hypha elements in fungal suspension, you will need to isolate the fungal spores with the filter or the glass wool. Basically, purpose here is to test the spores to preserve the spore properties in the testing, and not include things like mycelia, which will affect the properties of the spores. And also the low titers desiccation impact. So this one is basically sometimes certain microorganisms such as the pseudomonas can during the drying phase of the testing, the viability can be affected. So when you have those kind of microorganisms, take note of the drying conditions so as not to affect the viability of those microorganisms. And also checking the viability of the culture and making sure no cross-contamination is present before the testing. So this is to make sure that you are testing the right microorganisms without any other contamination present. Next. So this is the diagram of the Aspergillus spores. So basically this is considered a filamentous fungi. You can see it's a conidia spores as well with the mycelia. Next slide. This is a more diagram, SEM diagram on Aspergillus bacillus. You can see it's a spiny spores. So these are the things that we want to test that can pose the worst case challenge for the disinfectants. Next. So next we come to the surface. So some of the common causes of the failures is also the surface condition. So surface type and conditions can have huge impact on the efficacy, because if your surface is damaged or is rough due to wear and tear as well, so those surface conditions can basically pose a challenge to the testing, because it is not easy for the disinfectants to come into contact with the microorganisms hiding in those crevices on the surface. So it's very important to have a good surface condition for the testing. So preparation of the surface prior to testing, autoclaving may not be acceptable for some surfaces, because some surface, if you autoclave, the surface may be damaged. And then the residues must be removed, and no rusting, pitting of surfaces. This one will create the surface roughness, which can impact the studies results. And also some surface pose a challenge as well during qualification studies, peeling after sterilization due to the sterilization issues not compatible with the substrates, and the surface tension as well. Certain surface create more surface tension, and this is also can create some issues during the testing process. Next. So these are examples on the autoclaving of the substrates, which can damage the surface. So this is the surface came out from the substrates. Next. And then this is a surface tension issue. So you put the disinfectant on the surface, so it basically not evenly spread out. So it's due to the surface tension on the surface. Next. And also surface sterilization issues as well. This one surface is damaged. And during the creation of the coupon on the right, there's some clamping on the coupon. So damage the coupon surface during the creation of the size of the coupon for the studies. So this will impact the result as well. Next. And this is also another case of the surface degradation issues here. See the surface is all damaged. So this may also impact the study's results. Next. These are also challenging due to surface grid and the surface finish as well. You can see that there's a grid on the surface. So this one is also very challenging to perform the coupon studies. Okay, next. So next, we come to the recovery phase. So typical surface recovery methods is basically swabs and direct inoculation of coupons into neutralizing media. Contact plates is rarely used. So if you have the direct inoculation of coupons into neutralizing media, you will need sterile coupons, because if the coupons is not sterile, you may have some contamination issues over there. And this may include the manual or automated dislodging of the bioburden from the surface into the solution. And for the bigger surface area, you may need to use stomacher bags for the recovery. So this may involve additional handling if your coupon size is big. And whichever recovery methods is used must be validated and verified. So some of the typical methods can involve sonication, vortexing. So vortexing is based on the ASTM E2197, and also the glass beads is the EN13697. So these are some typical recovery methods can be used. And the final plates must be countable to calculate the log reduction. So there's a countable range. I think it's between 30 and 300. Make sure that it's within that range. Next. So these are some of the disinfectant qualification study tips. As mentioned before, AOAC test methods are not appropriate because it is only a pass-fail test. But some of the steps or procedures such as inoculant preparation, spore preparations can be used and is useful as well. And the most commonly used guidance documents is EN13697 and ASTM E2197. So these two coupon study guidance documents provide a very detailed steps on to guide you how to perform the coupon studies. So it's very valuable steps stated in the guidance document. And USP 1072 and PDA Technical Report 70 is useful in determining the log reduction, especially 1072. I mean, the microorganism, the acceptance criteria mentioned is at least 3 log reduction, and for the spores is at least 2 log reduction. And upfront proactive planning is extremely important for any coupon studies. Make sure that you plan what you want to do, evaluate the surface and then the microorganisms, and what are the disinfectants to be used, and how are you going to apply the disinfectants in the actual clean room. So all this will need to be carefully considered and planned in advance. And combining physical removal and chemical kills in one study is not recommended because physical removals can introduce some kind of variability into the study. So typically, this is separated based on the chemical kill. Consistency is crucial to a positive outcome. So these coupon study methods can have some variability, as in any micro methods, and you need to have a consistent methods and practices to ensure consistent results from the studies. And of course, reading the product labels to understand the product claims and limitations is necessary. Make sure that you understand what the disinfectants and sporicidal agents is effective, whether they're effective against spores or fungal spores or certain viruses and things like that. So you will need to understand before you proceed to the coupon studies. Expiry testing can be included as well, including the dilution, expiry dating, and also the shelf life. So this can be included if required. Of course, if you use the contract lab to perform the coupon studies, of course, this will reduce the amount of time and efforts from the end user, but you will need to basically work closely together with those contract lab to make sure that what they perform is according to your study plan, and also understand the limitations as well. Auditing of the lab is very useful also. Yeah. Next slide. So this is the case study on the substrate. So you can see here the different substrates on the left, different surfaces: stainless steel, glass, aluminum, epoxy. So in any clean room, there may be a lot of different kind of substrates. So typically you will need to perform some risk assessment to select the most representative substrates, because if not, you will have a lot of surfaces which can create a lot of tests for the coupon studies. And of course, there are different microorganisms to be tested: staph, Pseudomonas, Candida albicans, the yeast, and then the Aspergillus brasiliensis. As you can see here, the most challenging microorganisms is the Aspergillus brasiliensis. You can see or less than 3 log reduction. So if challenging microorganisms such as Aspergillus brasiliensis, you can see that it is challenging no matter what surfaces it is tested on. So typically this microorganism is used in coupon studies as one of the worst case challenge for the disinfectants. And then also you have the penicillium as well, another mole. This is easier to kill than the Aspergillus brasiliensis, but it also poses some challenges on some of the surfaces, as you can see here. So the surfaces selected is important because it can affect the efficacy of the disinfectants based on the type of surface and the type of the microorganisms. Next. So the keys to the successful qualification is basically having an effective antimicrobial agent, understand what is the active and what are the label claims on the disinfectants and the sporicidal agent, and having an effective and repeatable testing protocol for the coupon studies. And also, how are you going to apply the disinfectants onto the clean room surfaces by having effective sanitization procedures. And after you have completed the coupon studies, you need to have an effective change control procedure in place to assess any changes in the cleaning procedures in future, so as to maintain the qualified status of the disinfectant validation. Next. So requalification. So any requalification required. So this is based on the review annually to assess the risk or whether any significant changes have occurred. So some of these changes can be due to, for example, if you do a renovation in your clean room, you introduce new surfaces or new equipment with a very unique kind of surfaces, then you will need to assess whether you will need to perform an additional coupon testing for that particular surfaces that you have introduced. And also, if you have a new bioburden appears at high levels or inherently resistant organisms appearing in your EM monitoring, so you may want to perform a study just to make sure that your disinfectants and the sporicidal agent is effective against this new bioburden that you have just discovered. Of course, they can be due to other factors such as the ineffective sanitization procedures or transfer procedures for the materials into clean rooms, or it can be due to some filter leakage as well. But the bottom line is you will need to make sure that first you need to make sure that your disinfectant is effective against this bioburden. And of course, if it is effective, then you may need to perform a deeper investigation to identify the root cause for this. Lastly is the evaluate the qualification, the disinfectant validation every 5 to 7 years to determine if any repeat testing is needed. Because sometimes the test procedures or the guidance documents may change or get updated. So in those cases, you will want to evaluate whether disinfectant qualified many years ago is still valid based on the older version of the guidance documents or the test methods. So this will need to be taken into consideration. Of course, no routine qualification required unless for the following 3 points mentioned here. Okay, next slide. I think, yeah, I'll hand over the time to Jim again to finish this off. Thank you.
Okay, thanks, Richard. Excellent job. You are the man. So I'm going to come home and, or come in rather, and finish off the talk, and then we'll take any questions. So let's talk here a little bit about the in situ field trials, a very critical element of the disinfectant validation that you're doing. Regulators really like to see this kind of data. So let me go to the next slide. We have a poll question coming in here. I can see it coming. There we go. So the question is, how often does your facility, a clean room facility, conduct in situ field trials? So how often do they conduct these field trial tests? Is it once a year, twice a year after shutdowns, or after each adverse event, such as a construction event, power outage, contamination event, or a shutdown, or never? Let us know. All right. Okay, so it looks like a lot of you never do it. 40%. That's interesting. Richard, we got some work to do here. 30% after each adverse event. That's good. 21% twice a year after shutdown, and 9% once a year. So the nevers, we hope to move you into these other categories. That's our goal. Thank you. Okay, so I'll move along here. So this actually comes from a couple of recent publications. Both Richard and myself published an article, I guess it was a couple years ago now, Richard, that looked at our phenolic disinfectants followed by a sporicide. We also got one out there with our quaternary ammonium and a sporicide, and I'm going to be publishing some new data here on a couple of different directions, one using the Annex 1 contamination control strategy called CCS, and the other one will be looking at using a quaternary ammonium twice followed by a sporicide. But essentially, we're all looking at the same thing. What is we refer to it as a triple clean. So how does that work, you might ask me? Well, we gather environmental monitoring data after a worst case event, let's say construction event, power failure, shutdown. That's T0, collect the EM data. And this is the first time I do this. So then let's say Richard applies the phenolic low pH phenol in the facility. Then my EM team comes in and collects T1 EM data after that to see what's still in the room. Then Richard comes in again, he applies the high pH phenol, right? Then our EM crew comes in again and takes the T2 data, right? That's the name of a good Arnold Schwarzenegger movie, Terminator 2. So we get that data. Then Richard comes in and he's going to nuke the place with the sporicide, the hydrogen peroxide peracetic acid. So the idea here is to kill everything except maybe a few little outliers, but it should almost be all zeros. So that's the T3 data, right? That's our new Terminator model where we've killed everything, hopefully, in that clean room. That's the goal. So you should see that data going, those number of hits going down, 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 down. And we look at where does the definition come in from triple clean. There's no unified definition in the industry, but what PDA's technical report number 70 does state is it says facilities should strongly consider having a special startup cleaning and disinfection program in place for shutdowns or when significant construction has been performed. So one of the things that I promote to you is the triple clean using a disinfectant twice, followed by the sporicide. So you might ask me, well, Jim, why do you do it this way? Because the disinfectant both cleans and disinfects the surface. And in the US, they register these disinfectants as one-step cleaner / disinfectants because the disinfectant has surfactants. That's a key step because you want a nice, clean, pristine surface in order to lay the sporicide on the surface and get complete kill. So here's an example of how it works. So at time 0, my clean room facility, I have a lot of contamination after construction. So a lot of little bacteria, lots of green mold, and lots of red spores everywhere. You can see Richard's going to have his work cut out for him here. Then the idea is to mop from the most critical location, the filling area, go work your way out. So going from this high criticality area and work your way out of the clean room toward the gowning room. You never go from the gowning room in because that's a dirty area. So the idea is applying disinfectant like so. We apply the phenol or the quat 2 times, right? After those 2 applications, what do I have left? Just a few little spores, right? A few little outlying spores everywhere. Then Richard comes in and nukes the place with the sporicide, the bleach, or the hydrogen peroxide, peroxyacetic acid, and voila, nothing left. That's the idea behind the triple clean. So let me give you some more background on triple cleaning. We published many articles on this and a PDA poster and a PDA book chapter. They are designed to be used after worst case events. My colleague in industry, a well-known consultant, Anne-Marie Dixon, also promotes a 9X clean, which is essentially a triple clean. And here's the way she does it, 3 days in a row. So that creates a lot of downtime. And she's agreed with me here recently that the triple clean I just walked you through, is just as effective. You can also do a fogging event, pre-clean with disinfectant and then fog with a sporicide. You can pre-clean with disinfectant and hit the area with VHP, which is vaporized hydrogen peroxide. You can get a similar result. So here is the clean room facility in South San Francisco at Genentech. These are all the quadrants in a new clean room. Post-construction, it looks like this, and this is a clean room installed that was brought over from Copenhagen. So here we took environmental monitoring, and as you can see, there's lots of organisms. The red represent fungal and bacterial spores. The green represent vegetative flora. Almost every quadrant has something in it. So in this triple cleaning, we decided because it's so dirty, we're going to come in 3 times with disinfectant. So low pH phenol here. Notice that all the green is gone. That's vegetative organisms. Then we come in with the high pH phenol here, and keep in mind these also are cleaners. So I'm removing some here. Very good data here. Then I'm coming in with the low pH phenol again. More organisms are removed. Then we send Richard in again to nuke the room with the sporicide. He's done that, and all my quadrants are free of microorganisms. Now you may not get completely free. Very often does that happen. But the idea is to see a steady reduction over time in that data. And that means you've got some pretty good data. We have some case studies that we published on that. So that's something we can share with you with both phenols and with quats. And I've got another one that I'm working on. And again, to summarize what we walked you through this afternoon, it's evening for me, but it's, I guess, morning for you, is we walked you through the efficacy testing methods. So what is seen in the US and Europe, what's expected for regulators. We talked a little bit about what regulators expect to see. We talked about the testing challenges, right? Richard got into that. He talked about how to get the organism in the right phase of growth, how to neutralize it, how to do recovery. He presented some case studies on efficacy data. And then I spoke about the field trials just now. So these are all the references we used. We're happy to share any of them with you that are not copyrighted. But these are really good references for you to have on file if you have a clean room facility and you are either doing validation or you're going to be doing it, completed it or already done it. And we want to thank you for your time today. We want to especially acknowledge all these folks here at the bottom of the screen for all their help with the slides and the scanning electron microscopy. It's always the people in R&D and micro that do a lot of this work behind the scenes to make things look great and make it look great again. So with that being said, Linda and Christina, we are open to questions.
Thank you, Jim. Thank you, Richard, for a fantastic presentation. We've got a short amount of time actually for questions. So I'm going to quickly run through, and this is going to be a bit of a quick fire for you guys. So Jong Hyuk Lim asks, I would like to ask whether spore-forming bacteria can survive in the non-sterile wiper.
Potentially. I've seen, it's been a few years now, but there was an outbreak in the U.S. of Bacillus cereus in wipers. So it is potentially possible if it's not sterile and it's not been irradiated, there is a possibility. It's not common, but it could happen.
Okay, thank you. Also, just a quick note, for those of you who've received a chat message from Christina, please can you reply and identify yourselves? We really do need to know who's attending these sessions, otherwise it will make it difficult for us in the future. So please do reply to that. The next question from someone unidentified, IPMSBS1, is how many data is required for in situ testing?
I think it really depends upon, and Richard can comment on this too, I think it depends on how often you maybe have a shutdown or how often you have an adverse event like a power outage, construction event on site. So many of the sites in the US, for example, in the Northeast, if they have several power outages because of the winter storms, they may gather data 3 or 4 times a year. But then there are several facilities in say the Southeast that if they have shut down twice a year, they gather the in situ field trial data 2 times a year. Go ahead, Richard, you can comment too.
Yeah, I agree. I mean, during those adverse events, I mean, plant shutdown and also the microbial excursion in certain clean room. And if you are asking about the number of data point, I mean, if you're talking about the sampling point, it can be the routine EM monitoring locations, and also you can incorporate additional swab location as well based on your risk assessment on the risk of each area, whether it's a high touch point and things like that. So those you can basically work out a study plan based on those considerations.
Thank you. Please do keep posting questions or unmute yourself if you want to ask a question. We will get to you, and also if it's not answered, we will be able to come back to you with some replies following the session. Smitha asks, hello, our facility uses glass vials and ampules with paper labels and these materials to bring to grade A and B rooms for the product processing. How should I make sure the material decontamination is effective in this case? If we spray with disinfectant sporicide, the integrity of the label is lost.
So I would, if there's an issue with applying, say, a liquid sporicide, maybe investigate other methods such as VHP, vaporized hydrogen peroxide. They make VHP chambers. There's even one site in the US that uses UV light and uses the UV light to get a 5-log reduction on essentially plastic bags with ampules and vials in the bags. You can look at so some alternative technologies like that if the liquid disinfectant or sporicide's causing problems. Go ahead, Richard. If you want to comment.
Oh, no, okay. Yeah, no, I got no comment. Yeah, I agree with you. Basically, if liquid disinfectant is not appropriate, then you will need to consider the VHP, the gaseous type of disinfection.
Christine asks, how would we choose the environmental isolates to be used?
So let me comment on that, and then Richard can add to it. I always recommend looking at a tape over time of your environmental monitoring data, look at your trending data, and I would choose your environmental isolates based on what is frequently occurring. So for example, maybe a couple of gram-positive cocci, what appears to be worst case. So you may have 1 or 2 bacillus, 1 or 2 mold spores that appear to be worst case that pop up a few times a year, and you want to add them to it. You'll also probably want to add 1 gram negative. If you have a yeast, you can add it. If not, I probably wouldn't add it. And some of the cell and gene therapy facilities now are actually going outside to contract labs to generate additional data against some of the viruses like lentivirus and even viruses similar to parvo. And Richard, go ahead if you want to comment.
No comment.
Okay, next question. Is coupon testing also required when we are already conducting a routine environmental monitoring?
So the 3 pillars of the disinfectant validation are the laboratory testing, so the coupon studies, the suspension studies. Another one is the field trial, which is the data I covered at the end. And the third one is the ongoing environmental monitoring and data trending. So if all you've got is the EM data, that's good. That's one important key aspect that regulators want. But then it is important over time to take a look at your EM data and come up with some environmental isolates to run this coupon study. And after a worst case event, maybe a power outage, construction event, or shutdown, to generate that field trial data. Richard?
Yeah, I completely agree with you. I mean, EM data is a good indication on the effectiveness. And also the coupon studies can also complement on the EM data by taking into consideration some of the special isolates that you can identify in the facilities.
Okay, next question from Prerna. Would like to check on solution preparation tank sanitizing. Usually these cleanings using sanitization agents are validated based on rinse sample, but no microorganism spiking involved. How do we define our disinfectant stroke sanitizing agent efficacy for such cases?
Richard, you can handle this one.
Yeah, this one, basically you can work with the detergent supplier and the sanitizing agent supplier. They can perform some studies on this. If you have certain microorganisms of interest, you can basically do studies on the particular microorganisms that can be found in the system. So this can be worked out and perform some kind of studies to understand the efficacy. And some supplier can also provide some ideas on the efficacy of the sanitizing agent use.
Okay, and Julie asks, how frequent will we do the validation, and is there an acceptance criteria in validating disinfectants?
Yeah, I can, let me start on this and then I'll hand it to you, Richard. So I would recommend doing your disinfectant validation, the coupon testing piece and the field trial, especially, let's say if you're starting up a new clean room facility, it may take you say 6 to 12 months to generate the coupon data, because you need to figure out what your environmental isolates are over that period of time. But the field trial data, you can probably do some of that initially when you start up the facility to do that triple cleaning. So you'll do this initially, and then on a yearly basis, you'll evaluate your EM data and see if you have a new organism show up that appears to be hard to kill or worst case. And if you haven't tested it, you would want to probably generate some coupon data on it. And then I'd say every 5 to 7 years, you want to take a look at all of your field trial data, coupon testing data, and suspension data, and make sure that it is standard the test of time, that the methods aren't antiquated and outdated, and that you have the correct level of log reduction, and the studies were run well, so that that data can stand up to regulatory scrutiny. So that, I think, is all a key thing. Richard, you want to add to this?
Yeah, I agree. So basically, the key is basically to do an annual review as well on the EM data to see any adverse trend or any microorganism that is hard to kill. Yeah. So in those cases, you will need to perform additional studies to ensure that your disinfectants are effective. And as mentioned before, it's basically every 5 to 7 years, make sure that your test methods and the validation that was done previously is still up to date and is still in line with the guidance documents and the regulatory requirements.
Thank you. Now, if you are posting questions, please continue to do so. We are logging them and we will come back to you with replies. And we have, I think, time for just one more question if we ask for your indulgence. Do we have to validate the in situ study for all clean grades area, grade A, B, C, D, or would it be sufficient to validate only in grade D?
So you're speaking about the field trial data, and my opinion on that is after a worst case event, or let's say construction or whatever, generally that affects more than one type of clean room. So it's usually maybe, let's say, an ISO 5 area and ISO 7 because you're going to have workers coming in and out of the area. Let's say if it's construction. So you'll be able to do more than one area when you generate that data. And it's really a good time to get that, that field trial data, because in a recent study that we did with the University of Tennessee's parenteral medication site in the US, their clean room site, they were going in and doing some construction in their filling area. They had construction crews and operators coming in and out of the ISO 7, ISO 8, so ISO 8, ISO 7, and ISO 5 area, which is grade A, B, C, and D, right? And it just created a tremendous opportunity to capture all that data. And it really is something Thomas Arista and the regulators in our industry look positively on. Richard?
Yeah, I think it's a good idea to include the different areas, I mean, the grade areas, grade A, B, C, D areas, because I mean, some of the surfaces in this clean room may be different. There are different clean room substrates, and also in terms of the preparation of the disinfectants may be different as well, because in the sterile areas you are using the sterile disinfectants and the sporicidal agent. So there are some differences in terms of the preparation and also the substrates sanitization procedure as well. So it's good to include at least in each of these areas. Yeah.
Thank you both. I know there were a lot of questions and there will be some follow-up involved with that. We'd like to thank you for your time and for sharing your expertise with us this morning, and for everyone for joining us for this session. The session has been recorded, and the link to the presentation as well as the recording will be sent to you as long as we can identify you, and the team can follow up with you directly if you have any further questions. With that, we'd also ask for your help to fill in the evaluation form, as we would appreciate your feedback and how we can improve in future. We look forward to more sessions led by STERIS in the near future. Please do watch out for those, and we look forward to seeing you all again. In the meantime, thanks for joining us, and we wish everyone a good rest of day, and for Jim, a good rest, and thank you for joining us this evening.
Thank you very much. Thanks, Linda.
Thank you.
Thanks,
Thank you.
Christina.
Thank you.
Take care, everybody.
Look forward to
Bye-bye.
seeing you in August.
Yeah, thank you.
Yeah.
You too. Bye.
Take
Bye-bye.
care.